In this lecture, I will discuss some additional resources that we processed to build the Harmonizome. The Project Achilles dataset contains measurements of the effect of thousands of single-gene knockdowns on the proliferation of hundreds of cancer cell lines. This dataset is useful for discovering which genes cancer cell lines depend upon for survival. Those genes could be candidates for targeted inhibition by a small molecule as a treatment for cancer type that the cell line mimics. The Project Achilles dataset is structured as a matrix with genes labeling the rows, cell lines labeling the columns, and rows of scores indicating the relative sensitivity of each cell line to a gene knockdown. As we've seen from previous examples, a matrix like this can be interpreted as a bipartite graph, where the rows, in this case genes, form one set of nodes, the columns, in this case cell lines, form the other set of nodes, and the matrix values define the weights of edges connecting genes to cell lines. Because the scores are a measure of relative sensitivity, we have positive and negative values, resulting in a bipartite graph that is signed, with negative edges indicating that a gene knockdown reduces the fitness of a cell line, and positive edges indicating that a gene knockdown increases fitness of a cell line. This heat map is a visualization of a bipartite graph derived from the Project Achilles dataset. Red regions of the heat map indicate cell lines with increased fitness following a gene knockdown, and blue regions indicate cell lines with decreased fitness. This data could be useful for suggesting targeted therapies, where for each cell line, the gene knockdowns that most potently inhibit proliferation are selected. Other datasets to be discussed later can be used in combination with the Achilles data to gain information about how to match targeted therapies to cancers. The BioGRID dataset reports physical interactions between proteins that have been detected using high-throughput or low-throughput detection techniques. Many protein-protein interaction databases exist, such as HPRD, INTACT, and MINT. Protein-protein interaction data are useful for understanding functional relationships between proteins. The BioGRID dataset is structured as a list of interacting protein-protein pairs. This defines an unweighted graph. However, because all nodes are of the same type, they're all proteins, it is not a bipartite graph, which connects nodes of different types. This heat map is a visualization of the BioGRID dataset. Proteins label the rows and columns and have the same order. The colored regions indicate interacting proteins. This information is useful for understanding which proteins work together. The Cancer Cell Line Encyclopedia datasets contain gene expression, copy number, mutation, and drug sensitivity measurements for cancer cell lines. Genomics of drug sensitivity in cancer is another source of such data. Taken together, these datasets can be used to match drug treatments to molecular diagnostics. This involves using machine learning models to detect signatures of differentially expressed genes or genomic aberrations that are highly predictive of cell death following a particular drug treatment. The CCLE gene expression dataset is structured as a matrix with genes labeling the rows, cancer cell lines labeling the columns, and gene expression levels filling the matrix. As we have seen from previous examples, a matrix like this can be interpreted as a bipartite graph, where the rows, in this case genes, form one set of nodes, the columns, in this case cell lines, form the other set of nodes, and the matrix values define the weights of edges connecting genes to cell lines. In this case, we are interested in thresholding the data to extract associations between genes and cell lines in which those genes are expressed unusually high or low. This heat map is a visualization of the bipartite graph derived from the CCLE dataset. Red regions of the heat map indicate cancer cell lines with higher gene expression than usual, and blue regions indicate cell lines with lower expression than usual. This information could be used in combination with the Project Achilles dataset to test if any patterns of differentially expressed genes are predictive of sensitivity of cell lines to gene knockdown. The Catalogue of Somatic Mutations in Cancer dataset reports gene mutations in cancer tissues and cancer cell lines. This data is useful for obtaining lists of candidate gene mutations that could be driving particular cancers. The COSMIC dataset is structured as a list of gene-cell line pairs. This already places it in the form of an unweighted bipartite graph. This heat map is a visualization of a bipartite graph derived from the COSMIC data. Genes labeled the rows, cancer cell lines label the columns, and colored regions of the heat map indicate which genes are mutated in which cancer cell lines. This heat map shows that some genes are very frequently mutated in cancer cell lines, whereas others are infrequently mutated. Similarly, some cancer cell lines harbor many gene mutations, whereas others have only a few. As discussed for the CCLE gene expression data, this COSMIC gene mutation data is also potentially informative of how cancer cells will respond to treatment. So the COSMIC data could be combined with the Achilles data that we discussed at the beginning of this lecture to test if any gene mutations are predictive of sensitivity to gene knockdown. The datasets discussed in this presentation can be accessed via the links shown. As mentioned in previous lectures, the links DCIC provides access via the Harmonizome to process datasets derived from these resources. We hope you find our approach to abstracting biological datasets to bipartite graphs and networks helpful for understanding how diverse types of biological data can be standardized for subsequent integrative analyses.
Woohoo!